Imagine opening a DNA report at your kitchen table. It looks familiar, a neat circle, a few colored bars, perhaps a map with lines reaching toward recent ancestors. But this report is not from a real company. It is a hypothetical composite, built only to ask a question, because the scientific evidence does not come from one app or one person's result. It comes from comparing ancient reference genomes with DNA sampled across living populations. And when you make that comparison, the timeline beneath your kitchen table suddenly drops through the floor. You may expect a story about grandparents, borders, and surnames. The real answer is far stranger. Some of the fragments being studied entered the ancestors of living people tens of thousands of years ago through encounters with human populations that later disappeared as distinct groups. Their DNA did not survive as a hidden second person inside you. It survives, where it survives at all, as scattered segments, copied, shuffled, shortened, and passed through generation after generation. That is the first warning worth carrying into the story. A percentage assigned to Neanderthal, derived, or Denisovan-related sequence is not a percentage of your person. It is an estimate of genomic segments matched by a model to ancient reference material. Think less of a pie chart dividing a human being, and more of torn pages whose likely source can sometimes be recognized. The pages are real evidence. The pie chart can become misleading very quickly. For most of the time Neanderthals were known to science, there were no genomic pages to read. 19th century researchers had bones, cave sites, tools, and anatomy. Those clues showed that another kind of human had lived in Ice Age Eurasia, but they could not settle exactly how those people were related to the ancestors of living humans. Similar bones could suggest relationship, difference, or both, and the fossils alone could not show whether separated populations had later exchanged genes. A branching family tree seemed natural, one branch here, another there, each separating cleanly into the past. Then, in 1997, researchers recovered mitochondrial DNA from the original Neanderthal specimen. It was a remarkable first genetic whisper, but mitochondrial DNA follows only one maternal line. It is a single thread, not the whole woven history of a population. The larger question, whether Neanderthals had contributed DNA across the genomes of living people, needed nuclear DNA, the enormous archive carried in the cell nucleus. In 2010, a draft Neanderthal nuclear genome changed the shape of the question. Researchers compared genetic variants and found that Neanderthals shared more derived variants with sampled present-day Eurasians than with sampled sub-Saharan Africans. This was not one curious gene or one modern family. It was a genome-wide imbalance across carefully chosen comparisons, tested against alternative histories. That asymmetry supported gene flow into ancestors of populations outside Africa. In plain language, some populations had separated, met again, and had children. The family tree had branches that reconnected. The same year brought an even stranger discovery. DNA from a tiny finger bone fragment in Denisova Cave did not fit neatly with either Neanderthals or living humans. It revealed a distinct archaic population, now called Denisovans. By 2012, scientists had produced a high-coverage genome from that small fragment. The sequence allowed far finer comparisons of Denisovan relationships, diversity, and genetic contributions to living populations. An entire population, almost invisible in the fossil record, had first become legible through molecules. How can researchers tell that a matching sequence reflects ancient interbreeding rather than coincidence? No single resemblance is enough. The case combines genome-wide patterns, linked stretches of variants that resemble an ancient reference, and results reproduced across samples and methods. Recombination acts like a paper shredder over generations, breaking long inherited blocks into shorter ones. Their pattern and length can be tested against models of population separation and contact. Researchers must also consider alternatives, including old shared variation, population structure, and reference bias. One individual made contact unmistakably direct. Denisova 11 carried a Neanderthal mother and a Denisovan father. That is what the genome establishes. It does not tell you what either parent thought, how they met, what their relationship meant, or how common such unions were across Eurasia. The circumstances are unknown. The scientific power of the find comes from resisting the urge to turn one bone fragment into an invented drama. Other genomes show that the broader history was not one meeting at one place. High-quality Neanderthal and Denisovan genomes reveal multiple directions of gene flow, while DNA in living populations points to more than one Neanderthal encounter and several Denisovan-related contacts. The dates of particular contacts are estimated from track lengths and demographic models, not read from a single fossil calendar, so exact timing shifts with assumptions about generations and recombination. Studies have found at least 2 distinguishable Denisovan-related pulses, and recent reviews synthesize evidence for at least 3. But Denisovan may cover deeply structured populations separated across enormous distances and periods. It is a useful label, not a census of one uniform people. The physical map has also changed. A mandible from the Tibetan Plateau was linked to Denisovans through ancient proteins in 2019. Then, in 2025, proteins identified Penghu 1, a male Denisovan mandible from Taiwan, extending direct molecular evidence into a warm, humid part of Eastern Asia. These finds do not draw a hard border around a Denisovan territory. They are points of evidence in a much larger geography, still reconstructed partly from DNA carried by living people. Harbin added something different in 2025, one near-complete cranium, at least 146,000 years old, associated with one Denisovan population. Proteins from the cranium clustered with Denisova 3, and Denisovan mitochondrial DNA was recovered separately from dental calculus on its teeth. That makes Harbin unusually important for connecting molecules with a nearly complete skull. But it is one individual associated with one Denisovan population, never a universal portrait of all Denisovans across their deep and structured history. Now return to the fragments in living genomes. In broad, carefully qualified terms, many people with substantial ancestry from populations outside Africa carry roughly 1 to 2% Neanderthal-derived sequence in a single genome. Some Papuan and other Oceanian populations carry several percent Denisovan-related ancestry, with the upper end often summarized as about 5%. Smaller Denisovan-related components occur in parts of East and South Asia. These are population patterns, not values that can be assigned to every person in a region. The old slogan that Africans have none is also wrong. A reference-free study detected small Neanderthal-related signals in every population examined, including studied African populations. Much of the African signal can be explained by later migration back into Africa, along with older gene flow from early modern humans into Neanderthals. Different African populations have different histories, and no single number can describe a continent. East Asian groups, meanwhile, often show somewhat more detectable Neanderthal ancestry than European groups, but the size and explanation of that difference remain model-dependent. Even the same modern DNA can yield a different estimate when the scientific flashlight changes. A Denisova Cave Neanderthal genome was closer to the population that mixed with ancestors of many living people than the Altai Neanderthal reference. In one analysis, using Denisova allowed researchers to detect 10 to 20% more Neanderthal sequence than with the Altai reference. That does not mean 10 to 20 percentage points more ancestry appeared. It means the reference helped reveal additional matching fragments. Samples, thresholds, comparison populations, demographic assumptions, and algorithms all move the estimate. Here's the counterintuitive twist. The fragments that remain are only part of the evidence. The missing fragments matter too. Maps of archaic ancestry show depleted regions near genes and on the X chromosome, and population genetic models suggest that selection removed substantial introgress material over time. Some surviving segments may be neutral. Many have unknown or unmeasured effects. Some have studied functions or became common enough to attract attention as possible adaptations. There is no supported reason to imagine that evolution simply saved the best gifts. The clearest classroom example is a Denisovan-related version of the EPAS-1 region in Tibetan populations. It shows strong evidence of selection and is associated with hemoglobin regulation under high-altitude, low-oxygen conditions. It contributed to population-level adaptation within a much larger physiological system. It is not a magic mountain gene, and it cannot predict how one individual will feel, perform, or remain healthy at altitude. A Neanderthal-derived haplotype around the OAS immune genes offers a different lesson. Experiments show effects on gene expression and splice forms involved in innate immune responses. That is a measurable molecular function. It is not proof of universally better immunity. A response can depend on the pathogen, environment, other genetic variants, and the health outcome being measured. Helpful in one context, harmful or negligible in another is not a contradiction. It is biology. The TBX15 and WARS2 region belongs one step lower on the certainty ladder. A deeply divergent Denisovan-related haplotype shows a selection signal and associations involving gene expression, DNA methylation, adipose differentiation, and body fat distribution. That makes it a candidate for archaic-assisted adaptation involving fat biology. It does not demonstrate a cold tolerance mechanism. The tempting story is more complete than the evidence, so the evidence has to set the boundary. This is where the modern mirror can distort the ancient one. A statistical association in a cohort is not proof that an archaic segment caused a modern outcome. A molecular effect is not automatically a benefit. A population pattern is not a diagnosis for one person, and ancient population labels must not be mapped onto modern racial categories. That is an editorial and ethical guardrail, while the empirical story underneath it is one of repeated migration, overlapping histories, and back migration, not pure lines moving through sealed boxes. The genome therefore cannot turn Neanderthals or Denisovans into rankings among living people, nor can it make these segments instructions for who you are. The overwhelming majority of any living genome comes through the long history of modern human populations, and every trait emerges from complicated interactions among many variants, development, environment, and experience. Archaic fragments are evidence of ancestry and population history. They are not verdicts delivered from the Pleistocene. There is one more reversal hiding in the numbers. No living person carries a complete Neanderthal or Denisovan genome. Different people carry different pieces. Recombination means one descendant may inherit a fragment that another does not, while drift and selection change which pieces remain common. Yet when researchers study many genomes together, those scattered pieces become something like a library distributed through a crowd. The vanished populations are not restored, and their full lives remain beyond genetic reconstruction. But part of the record of their encounters can be assembled from fragments that survived in different descendants. So the clean tree becomes a braid. Populations separated, changed, moved, met, and separated again. Neanderthals and Denisovan populations eventually disappeared as distinct groups, while modern human populations continued carrying some of the genetic consequences of those meetings. The lesson is not that ancient DNA divides living humanity into older boxes. It is that human history was connected even when its populations were different, and that disappearance did not always mean leaving nothing behind. You began at a kitchen table, looking at a hypothetical report made of tidy colors and percentages. Now the paper looks different. It is not a verdict about you, and it is not a set of borders around your family. It is a rough map of encounters, drawn from ancient reference genomes and patterns across living populations. The circle and bars were neat. The human backstory beneath them was never neat, and some of the people missing from the picture are still present in its scattered pages.
Thanks for watching this video. Don't forget to subscribe